What is DNA replication? What is DNA replication? Tell me, what is DNA replication? So I have written DNA replication. Replication means to form replica. Replica means you can say a photocopy. How will you compare? You have a paper, you put it in the Xerox machine and you get another copy. It is replica of that original copy, right? In the same manner, DNA replication. Replication means to form new copies of DNA from parent DNA, right? Similarly, in the Xerox machine, when you put A4 page and you get a replica, in the same manner, from a parent DNA, you form another copy of DNA. It is known as DNA replication. Any doubts? Replication means to form replica, right? To form new DNA from parent DNA or daughter DNA from parent DNA is known as DNA replication, right? And when you synthesize RNA, it is known as transcription. From DNA, when you synthesize RNA, it is known as transcription. And from RNA, when you synthesize proteins, it is known as translation, right? So today's class is about DNA replication, right? We'll discuss it in prokaryotes. Tomorrow's class will be on eukaryotes. Today we'll discuss prokaryotes in complete, right? So replication is to form replicas, right? To form new copy of DNA from parent DNA, right? So we'll discuss now the several salient features of replication. Salient features of replication, right? The first feature is that it is semi-conservative. What does it mean? Semi-conservative. We are doing replication, right? That means we are forming daughter DNAs from parent DNA, right? And the first feature is that it is semi-conservative. Semi means half, right? Conservative means it is preserved. During formation of copies, half of the original DNA is conserved, right? Semi-conservative means half, during formation of copies or replicas of DNA, half of the original DNA is conserved, right? How will you understand it? Suppose we have a DNA strand, right? And it has one strand in blue color and other is in green color, right? So we have a double stranded DNA, right? And we have two strands. One is our blue strand and other is our green strand, right? Clear or not? Right? And we are forming new copies of DNA, right? Since we are forming new copies of DNA, the first rule is that it is semi-conservative, right? It means half of the original DNA is conserved. How? See, One strand is green and one is blue. In the new copies, now see, this was the original DNA strand, right? One strand was blue in color, another was green in color. During formation of copies, one of semi-conservative, one half of the original DNA is conserved. In new copies formed, one this and one this. One contains blue strand and one contains green strand. And the new strand formed is this black one. In this also black one, but from the original DNA we got one green strand here, and here we got our blue strand, right? So the first rule is clear. During replication, that means forming daughter copies from parent DNA, half of the original DNA is always conserved. This is the first rule of DNA replication. For how? Because we made two strands, one was green in color and one was blue in color, right? During formation of copies, half of the strand was conserved in both the two copies. We got green strand here and the blue here, and the rest new strand was found, that was our black one. So right, so replication is done now. We moved from original DNA to two copies. We did replicas of DNA, right? But in this, one half of the original strand is conserved. How? Here green is there, here blue is there. So half of the original DNA is conserved during replication. So this is known as semi-conservative rule of DNA replication. Clear to all of you, or any doubts are left? Right? First rule is covered. Now coming on to our second rule. And one more thing, this was proved by Meselson-Stahl and Stahl's experiment. Right, the first rule was proved by the scientists Meselson-Stahl, that one half of the original DNA is conserved, the semi-conservative rule of DNA replication. Right, now coming on to the second rule for replication is that comes on the point of origin of replication. Today we are doing replication in prokaryotes. Tomorrow we'll be doing replication in eukaryotes, right? So coming on to the second rule about origin of replication, right? There is difference between origin of replication in prokaryotes and eukaryotes, right? In prokaryotes, the mechanism is a bit different from eukaryotes. What are the major differences? First reason is that in prokaryotes, there is a single origin of replication. Right? In case of eukaryotes, there are multiple origins of replication. Clear? The first rule was DNA replication is semi-conservative, that means half of the original DNA is conserved. Now coming on to origin of replication, the origin is different in prokaryotes and eukaryotes. In case of prokaryotes, there is only single origin point of replication, and in case of eukaryotes, there are multiple regions of replication, right? And now, the point at which replication start is highly rich in AT-rich base pairs. Suppose we have a DNA strand 5 prime to 3 prime and 3 prime to 5 prime, right? The strands are anti-parallel, right? Suppose we are starting the replication, that is origin of replication, right? And this point is highly rich in AT-rich base pairs, right? So first rule was semi-conservative. Half of the original DNA is conserved. Original replication, there are some differences between prokaryotes and eukaryotes. In prokaryotes, there is a single origin of replication. In eukaryotes, there are multiple regions of replication, right? And the point of original replication is highly rich in AT-rich base pairs, adenine and thymine-rich base pairs. At that point, the replication starts, right? And moreover, one more thing. At the point of origin of replication, there is a DNA A protein, right? And what is its function? It has 20 to 50 monomeric units, right? The first point was semi-conservative. Half of the original DNA is conserved. Suppose we have one strand is blue in color, another is black in color. During replication, one of the original DNA is conserved, right? It was proved by Meselson-Stahl Experiment. Second is origin of replication. The origin of replication, the point at which origin of replication starts, right? Origin of replication is highly rich in AT-rich base pairs, right? And during the point of origin of replication, there is a DNA A protein attached that contains 20 to 50 monomeric units, right? And moreover, there is difference between prokaryotes and eukaryotes. Origin of replication. Prokaryotes, there is a single origin of point. There is only one point. But in case of eukaryotes, there are multiple points, right? And the point at which it starts, it is highly rich in AT-rich base pairs. It contains high content of adenine and thymine base pairs, right? Now, the third rule is that, coming on to the replication bubble. What do you understand from bubble? What do you understand from bubble? Bubble. And suppose this is a strand and this is a strand. 5 prime to 3 prime and 3 prime to 5 prime, right? The two strands, one is 5 prime to 3 prime, another is 3 prime to 5 prime. Now, when that strand there is a formation of bubble where replication starts, right? There is difference in prokaryotes and eukaryotes, right? Thing is that this is normal 2 strands of DNA. In case of prokaryotes, there is a single replication bubble formed. It is like a bubble. When two strands are separating, it forms like a bubble. And in case of prokaryotes, there is a single replication bubble is formed. And in case of eukaryotes, what happens? Five prime to 3 prime and 3 prime to 5 prime, right? So there are multiple replication bubbles. Clear to all of you or not? We did DNA replication. Replication means to form replicas or xerox of DNA from the parent DNA or the A4 page we started, right? And replication has some certain rules that by which it is governed. The first rule is semi-conservative. That means half of the original DNA is conserved during the formation of new or daughter DNA, right? Original replication I discussed, it is highly rich in AT-rich base pairs, right? And moreover, there is a single original replication in prokaryotes, and in eukaryotes there are multiple regions of replication, right? Coming on to the third point, that is our replication bubble. Bubble means when the two strands separate, they form a bubble-like appearance. We have a strand 5' to 3' and 3' to 5'. So here a bubble is formed, but in case of prokaryotes there is single bubble is formed. In case of eukaryotes there are multiple bubbles are formed, right? Why? I told you in prokaryotes there is a single origin, so single bubble. In eukaryotes there is a multiple origin, so multiple bubbles. Clear? So 3 points are covered now. Fourth is our RNA primers. What is RNA primer? Tell me what is RNA primer? This is a normal strand of DNA, 5' to 3' and 3' to 5', right? So when we unwind the strands of DNA, please don't talk. Anybody who are talking, they are honored to leave the class. They are not supposed to sit here. They can get attendance and get lost. I already told, please don't disturb me. Okay, so 5' to 3' and 3' to 5'. Now we are coming on to the RNA primer, right? Here we are synthesizing DNA, right? Right? So there is a point here that is known as RNA primer, right? So on the leading strand, when we synthesize DNA, new DNA, right? The synthesis starts from a point that is known as RNA primer. We need a primer to synthesize multiple copies. In the same manner, when we do photostat or xerox, we need A4 page on which the another copy is formed. In the same manner, to form new DNA, we need a small copy of RNA known as RNA primer. And how many base pairs it is having? How many base pairs? It is having 5 to 50 nucleotides. This RNA primer is having 5 to 50 nucleotides, right? So let's make it in black color. Right? So when we have a RNA primer that is having 5 to 50 nucleotides, on this black molecule, the new DNA is synthesized, right? Because we are forming copies of DNA. That is known as replication, right? So new DNA is being formed on this primer. So we need a RNA primer to synthesize new DNA, right? So I repeat, conservative semi, half of the original DNA is conserved. Original replication, there is difference between prokaryotes and eukaryotes. Prokaryotes single origin, eukaryotes multiple origins, right? In the same way, replication bubbles, prokaryotes single origin was there, so there is single bubble is formed. In case of eukaryotes, multiple origins are there, so multiple bubbles are formed, right? Coming on to the RNA primer, to synthesize new DNA, or we are comparing it with a photo machine, photocopy Xerox machine, right? So to synthesize, do a photocopy, you need A4 page and then you make a copy. In the same manner, to synthesize new DNA, you need some all RNA primer that contains 5 to 50 nucleotides. On this, the further DNA is formed, right? Up to this it is okay, right? And moreover, I will cover some more points after that. And one more point, that is bidirectional. What does mean bidirectional? What is the meaning of bidirectional? That means it is on two sides. Suppose this is a highway, national highway, and one is here is New Delhi, and here is Amritsar. Right? There is a highway, you are going from Amritsar to New Delhi, right? And what will happen? The roads are anti-parallel to each other. There is a divider inside, right? But on one side cars are going this side, and other sides cars are coming this side, from New Delhi to Amritsar and Amritsar to New Delhi, right? So it is bidirectional. Right? In the same manner, on the two strands, that is 5' to 3' and 3' to 5'. It is, the synthesis is going bidirectional. Bidirectional means both the strands opposite side, but it is going on in two sides. Bidirectional, but opposite direction. Right? Semi-conservative, half of the original DNA is conserved. Origin of replication, multiple origin of replication in eukaryotes, single origin of replication in prokaryotes. And replication bubble is formed where two strands unwind. In case of prokaryotes, there is a single bubble formed. In case of eukaryotes, multiple bubbles are formed. RNA primer is a photostat machine on which further DNA copies are synthesized, right? So to synthesize new copy, you need a RNA primer that contains 5 to 50 nucleotides on which further DNA is made, right? Next point is bidirectional. On a highway from New Delhi to Amritsar, one road is on this side, another is on this side, right? So it is bidirectional, on both sides synthesis is going on, but in opposite direction, right? So we covered the 5 features of this replication, right? Now coming on to the next point, bidirection is okay to you or not? And now coming on to one more rule, what is the role of DNA helicase? Sixth rule is role of helicases. Helicase means, what does helicase means? Helicase means to unwind. Helicase means to unwind. Suppose this is strand 5' to 3' and this is strand 3' to 5', right? I told that when the strands separate, a bubble is formed, replication bubble, right? And for the formation of replication bubble, we need an enzyme known as DNA helicase. I am making it in black color. This is a DNA helicase. Helicase means unbound, it will open. How will it operate? It like a zip opener, right? So we have two strands of DNA, they are parallel to each other, they are stacked very well, right? When it will start moving inside, it will start opening the two DNA strands like a zip, right? So it will keep on moving inside, and the strands will start opening like this. The strands too will separate. So DNA helicase is a unwinder or zip opener. We have a zip, to unwind it, the two steps open. In the same manner, when DNA helicase is run between the two DNAs, it will move inside and open the two strands, and it acts as a zip opener. Clear? Up to this. Semi-conservative means half of the original DNA is conserved. It was proved by Meselson-Stahl Experiment. Origin of replication, coming on to that point, there are multiple regions in eukaryotes and a single region in prokaryotes, and this origin of replication is rich in 80 rich base pairs. 80 rich base pairs are rich in it, And coming on bubble, when the two strands are opened, a bubble is formed, and in case of prokaryotes, a single bubble is formed. In case of eukaryote, there are multiple bubbles are formed. RNA primer, on which DNA synthesis takes place. It is written 5 to 50 nucleotides, on which further DNA copies are made on the complementary strand. And one more thing, bidirectional. On the both strands, it occurs in opposite direction. Bidirectional means both sides, right? Helicases, it is a DNA helicase. It is an enzyme that unwinds the two strands of DNA like a zip. So it is a black color. It is moving inside, and the strands are opening and unwinding, right? Now coming on to the next point. I told the two strands are complementary, 3' to 5', 5' to 3', right? Opposite direction are there, bidirectional, and this was our helicase. Helicase means it has to unbind the two DNA strands. When helicase will move in like a zip, it will open the strands. So it will elongate and it will elongate, and it will move inside, and the whole DNA will open for the replication, right? So this is helicase, right? Now we have this single-stranded DNA and this DNA. On the outer side of this, these green ones are SSBs. SSB. Single-stranded DNA stabilizing proteins, SSB. These greens will stabilize the single-stranded DNA so that they don't break up or get attacked by nucleases, right? Normally, DNA is intact, 5' to 3' on one side and 3' to 5' on other side, right? Now we had opened the DNA. What is the zip opener? That is our helicase or unbinder. It is opening the zip. Now the two strands are separated, but these two strands are unstable, and they are prone to attack by some antioxidants, oxidants or enzymes like nucleases. To prevent this, there is green. This green are shown by single-stranded DNA binding protein, stabilizing protein. They stabilize the single DNA, right? First, the two strands are there in which helicase is, so the two strands are separating, right? When the strands are separated, the single strand is unstable for replication. To stabilize it, we attack SSB, single-stranded DNA binding proteins on the outer side. They will stabilize the single strand of DNA, right? And after that, and I am supposed to give you one mnemonic for you. Forget the directions 3' to 5', on which direction it is going, 5' to 3'. Multiple choices come in +1, 2. Result आ गया किसी के यार का? चलो, just discuss that first. right? These are the two strands. They are anti-parallel, right? One is this peach colored and this is green colored. This is in 3' to 5' and this is 3' to. They are anti-parallel. Clear? Now you move bit back. I told you that I lost my DNA helicase. Yes. थोड़ा पीछे हो जाओ, थोड़ा उधर हो जाएगा, मुझे मूव करना ना बीच में. Now you will open like this, right? This is 5' to 3' and this is, so I am this green Y is like DNA helicase, right? Now these strands are tight, tight, right? So when I will move, the strands will like start opening like a, no, you will remain like this, but it will open like a zip, right? I am DNA helicase. Clear? This strand was 5' to 3' and this was 3' to 5'. Both these strands are anti-parallel to each other, right? In case of prokaryotes, single origin of replication. In case of eukaryotes, multiple origins of replication. First point is that these two strands are supposed to be unmined, right? So first comes the role of DNA helicase. Then I will start moving inside like a zip opener, the strands will open, right? Now come one student, one more. Wilson, come. इधर गिरना जाए. So Wilson is our DNA helicase. It is unwinding the strands. Now I told that the strands are unwinded. So single strand is unstable, right? दो बच्चे और आओ. दो बच्चे आ जाओ. यार इतना हेजिटेट क्यों करते हो? तुम जल्दी आ जाओ. जल्दी आ जाओ. So hold hands together. Right. So it is attached on the outer side of the single strand, SSB. It is stabilizing this single DNA. Right. In the same manner, Kumud is attached on the outer side of this DNA. Here. So this is our SSB. Right. It is stabilizing this strand and he is stabilizing this strand because the two strands are unwinded. Right. First was the role of DNA helicase. She was moving inside and the strands were opening, right? Now the strands are open. This strand is unstable and this strand is unstable. Here comes their role. They will attach hands and stabilize this single DNA. And similarly, she will attach hand with this strand and the single strand is stabilized. This is the role of SSB, single stranded stabilizing proteins. Now the strands are unwinded and the single strand is stable. Now comes the role of RNA or DNA polymerase, RNA primer. One more. This is the leading strand and now we are attaching here RNA primer. RNA primer will do what? It will synthesize the further copies of DNA. Come. The two strands are separated and RNA primer is attached here. Right? Outer side is SSB. Outer side is SSB. The two strands were separated. Helicase has done its role. The two strands are separated. The single strands are stabilized by SSBs. Clear up to this? Now we have to synthesize DNA. It is synthesized on this. She is our photostat machine. A4 page is there, but electricity has come via RNA primer. Now I am synthesizing the DNA. In which direction? Synthesis happens in 5' to 3' direction. So on 3' side we are adding nucleotides. And I have now DNA polymerase 3 with me. I am attached to RNA primer. Now I will synthesize DNA in this direction. Clear to all of you or not? Are you able to understand? First, the role was for Wilson. अब रुको, उधर चलो. First came Wilson, right? What she did? अभी बाहर चलो तुम सारे. First came our, show them. First came helicase, the two strands. तुम भी रुको, SSB दोनों पास से हट जाओ, अभी अलग हो जाओ. SSB रुक जा, अभी बाहर आ जा. First the two strands were intact, 5 prime to 3 prime and 3 prime to 5 prime. First comes Wilson. Wilson has opened the zip. Right? So the two strands are separated. Then comes our two single-stranded DNA. He will join hands with this girl and stabilizes the single strand. And it is attached on the outer side. Similarly, Kumud will come and stabilize the single strand. Now the two strands are stabilized and they are ready for replication. Now comes our RNA primer on which synthesis will happen. Then comes the role of DNA polymerase 3. I will join hands and we are going to synthesize the DNA. Are we able to remember it now? Now we have synthesized DNA up to this point. Now what will you do? We have to stop the replication. To stop it, first I was synthesizing DNA by attaching to the RNA primer. Now one more student comes. Jaldi aata, jaldi. Jaldi, jaldi aao. Jaldi. Ek aa jao, jaldi. To stop here, what will come? Our DNA polymerase 1. So we were synthesizing DNA, but now comes this green girl. That is our DNA polymerase 1. I was synthesizing the DNA with her. At some point I have to stop it. Here comes this DNA polymerase 1. At this time I will lose her, and here the DNA synthesis is stopped. Up to this it is okay now. Now coming on to this lagging strand, right? This was from, इन्होंने उल्टा बनाया, थोड़ा बाद में करते हैं ना, दी चाहिए था ना लैगिंग वाला. तो डायरेक्शन वाज 5 prime to 3 prime, 5 prime to 3 prime. On this strand the synthesis is discontinuous. Why? Because DNA polymerase 3, which runs on this RNA primer, runs in 3 directions. So here the direction is 5 prime. So here I have to synthesize in this direction, because 3 will move in this direction. No? On 3 prime side we are supposed to add these nucleotides, right? Now just come, बाकी जाओ और थोड़े 3-4 रुक जाओ, रुको, वो छोड़ दो बाकी सारे. हाँ. Now we have to show semi discontinuous replication. पकड़ो अच्छी तरह, सिंगल शॉट में पकड़ो, पकड़ो. तुम इनके पैरेलल आ जाओ. अभी स्ट्रेट लाइन में. हाँ. तुम इनके पैरेलल आओ ना. कम नहीं, इधर इधर इधर. तुम थोड़ा कम हो जाओ, डिस्टेंस कम करो. Yes. Now I'm forming lagging strand. Don't remember the colors, just try to imagine the concept. On lagging strand there is, hold them tightly, there is discontinuous synthesis. One strand forms small copy and one strand forms small copy. Now comes the role of, these two strands are separate. Now I will join hands with them and ligate this strand, and a complete copy is formed. DNA ligase. Ligase is a ligator. It will attach these separate DNAs together. I think the concept is easy to you now, right? And one more thing, last point. Show me 2 strands. आओ, पैरेलल आओ, छोड़ दो. अटैच करो दोनों DNA आपस में बाइंड करो. First point was DNA helicase. It will unwind the strands. When I will move the strand, आपने इसको टाइट नहीं करना. When I will unwind them, there will be formation of supercoils on this side, right? Right. छोड़ो छोड़ो छोड़ो, थोड़ा slow. So there will be formation of supercoils on this side. These supercoils are overcome by topoisomerases. DNA topoisomerases. Right? I think it is easy for you now to remember the enzymes, the concept. First, there were two strands. The strands were unbinded by helicase. Then there was a single strand and there was a single strand. They were unstable. They were stabilized by green ones, SSB on this side and SSB on this side. Right? Then came RNA primer inside, on which DNA synthesis was supposed to be done. And DNA polymerase 3 was used and synthesis happened. Right? And to stop it, you use DNA polymerase 1. Right? It is done. And on the lagging strand, it is semi-discontinuous because it is formed on the 3' side. Right? To attach the separate strands, you use DNA ligase. Ligase means ligates. So two separate strands are attached by a DNA ligase. Right? And the problem of supercoils was overcome by, when supercoils are formed, it is overcome by DNA topoisomerase. Right? Thank you.